In the cell cycle, interphase is the longest process, which includes G1, S, and G2 phases. Cells that are non-dividing can remain in interphase stage indefinitely by entering the G0 stage. Mitosis and cytokinesis are relatively short processes of cell division. Interphase is the stage where cell growth and copying of chromosomes in preparation for cell division occur. Cells do not divide in interphase. The first two stages of interphase are G1 and S, which can be shown here. G1 phase is where chromosomes are decondensed. Chromosomes are replicated during the S phase. Here, the orange represents the centrosomes, the green represents the nuclear envelope, and the pink represents the chromatin. The G2 phase is the second growth phase in mitosis. In this phase, spindle formation begins, which can be shown here in purple. This is the prophase stage. In this stage, the nucleolus disappears and the chromosomes condense into discrete structures of chromosomes, which can be shown here in pink and yellow. These chromosomes are made up of two sister chromatids joined at the centromere, which is shown in black. Mitotic spindle forms as centrosomes move apart. This is metaphase. In this phase, centrosomes are positioned at opposite poles of the cell, and chromosomes line up along the central position in the cell called the metaphase plate. This is anaphase. In this phase, each chromatid is moved to opposite poles, and the chromatids separate. Poles of the cells tend to move apart in this stage. This is telophase. In this stage, chromosomes have migrated completely to the poles. Nuclear envelopes reform around the chromosomes. Chromosomes decondense and the nucleolus reappears. Cytokinesis begins completing cell division in this phase, and chromosomes decondense. Microfilaments are responsible for the process of this stage. Microfilaments form a cleavage furrow near the metaphase plate, which pinches the parental cell into two daughter cells. The division of a diploid cell by mitosis produces two cells which contain identical sets of chromosomes. The number of chromosomes by the end of mitosis remain the same. The process of mitosis is what gives our cells the ability to replace themselves. This process of replacement of cells happens in our bodies all the time. Humans would not be able to survive if our cells could not heal from illnesses, viruses, or injuries. The process of meiosis begins with meiosis 1, which separates homologous chromosomes. In interphase, chromosomes duplicate and the cell grows. This is prophase 1. In this stage, crossing over occurs where a pair of homologous chromosomes line up closely together and a tetrad is formed. The site of crossing over is called the chiasmata. Similar to the mitosis process, the centrioles move away from one another and the nuclear envelope breaks down. This is metaphase 1. In this stage, tetrads line up at the metaphase plate. This is anaphase 1. In this stage, pairs of homologous chromosomes split up and the chromosomes move to opposite cell poles. The homologous chromosomes move to opposite poles to separate, yet the sister chromatids remain together. This is telophase 1. In this stage, the spindles continue to move the homologous chromosomes to the poles. Once movement is complete, cytokinesis 2 occurs, where each pole has a haploid number of chromosomes. At the end of telophase 1 and cytokinesis, two daughter cells are produced, each with 1/2 the number of chromosomes of the original parent cell. Meiosis 2 separates sister chromatids. Meiosis 2 begins with prophase 2, where the nuclear membrane and nuclei break up while the spindle network reappears. This is metaphase 2. In this phase, the chromosomes line up at the metaphase plate. This is anaphase 2. In this phase, the sister chromatids separate and move toward the opposite cell poles. This is telophase 2. In telophase 2, distinct nuclei form at the opposite poles. Cytokinesis is the final stage of meiosis. At the end of meiosis 2, there are 4 daughter cells, each with 1/2 the number of chromosomes of the original parent cell. Meiosis results in an offspring which has the genetic material of 2 separate individuals. On the other hand, mitosis has no sharing of material but instead only division of cells.